Okay, so hi everyone. In case you're new here, I'm Makriti, and I'm one of the community editors for the PLOS Synthetic Biology community. And today I have the pleasure to be joined by Dr. Tom Ellis, Dr. Guy Stan, Dr. Francesca Cerrone, and as well as Reese Algar from Imperial College London. They're all researchers in the bioengineering department. And specifically, we'll be talking about a paper that they just published called Quantifying Cellular Capacity Identifies Gene Expression Designs with Reduced Burdens. And in essence, what this paper talked about was a technique that they developed in order to quantify the burden that cells are under when we impose synthetic constructs into them. But I'll let them talk more about what they did, the aims of their research, and how they kind of came up to this particular technique. So whoever wants to start talking first, you can go ahead.
I'll begin then. So just to give a little bit of back history on the project, what we did was both Guy and myself were starting our PI positions at the Synthetic Biology Center here at Imperial at the end of 2009, beginning of 2010. And we had to think of a project that a project student could take on, kind of a master's student before for about a 9 months. And Guy and I were inspired by a paper from Ling Chong Yu's group, who'd shown that because a circuit they'd designed put a stress on the cell that slowed its growth down, the circuit then performed in a way unexpected, where they got this sort of bistability, which could only be explained by the feedback loop provided by this decreasing growth rate. So me and Guy met up in Cambridge, because we were both there at the time, over a coffee, and scoped out a project, which then Reese took on as his master's project. It was like a 9-month project with me and Guy co-supervising Reese. Kind of a nice project to begin with, because it's a bit of lab work and a bit of mathematical modeling thrown in. And Reese comes from a mathematical background, and so does Guy, and I supervise a lab doing more experimental work. And so that project was initially sort of saying, well, there's lots of different things you can change with constructs, like the strengths of the promoters, the strengths of the ribosome binding sites. And maybe if you change quite a few of them, you could get a construct that you're designing, oscillator or a feedback system, to still work the same, but maybe different designs would give different amounts of burden, like so the growth would change. So Reese kind of followed that up to begin with, and then after the sort of 9 months was up, we got the funding for him to continue it on during his PhD. And about similarly, a few months into his PhD, the project kind of evolved as we spotted that you could also get a handle on how much burden things are producing by looking at something which is completely not connected to the circuit you're putting in. And so I think maybe Reese, you could chip in about how the sort of the burden monitor arose during the project.
Yeah, sure. So as Tom pointed out, we were originally just sort of concentrating on the actual circuit itself and the growth rate. So we spent a while trying to look at some feed-forward loops. So I think a lot of that time was spent me learning how to clone, to be honest. But yeah, so I mean, what we decided, well, what we really sort of decided we want to start thinking about was sort of the global impact of a synthetic circuit, and we were really trying to identify, okay, how can we strip out all of the other factors? So let's not think about toxicity. Let's not think about regulatory crosstalk. How can we look at how a synthetic circuit impacts upon a cell? And so the obvious way that happens is through the sort of shared resources. So we spent a while sort of thinking about how the best way to interrogate these shared resources was. There are a few papers out there, some of which saying that ribosomes are the key bottleneck. Some saying it's definitely not ribosomes. So we try to think of the most simple way that we can interrogate how a synthetic circuit was affecting the cell through these shared resources. We wanted something that was going to be able to be used across lots of different laboratories, didn't require any specific or special equipment, and just a plate reader was all that was needed. And so, we came up with this design for sort of this burden monitor, which was purely synthetic. So we had absolutely no native.
Oh no.
From the very simple constitutively expressed GFP that we tested, we wanted to see if we could design it onto plasmids. So we put it on a high copy, a medium copy, a low copy, and genomically. And we ended up using a genomic version for a number of reasons. And so, yeah, and at that point, we developed this constitutively expressed GFP, purely synthetic circuit that we'd integrated into the genome. And so at that point, we thought, right, we hypothesized this circuit should be able to tell us how much we're impacting on the cell's shared resources. So the idea being that the more GFP being produced, the more cellular resources that are available, and if that amount of GFP dims, then we're using more cellular resources. So essentially that was, I think, the process of coming up with the burden monitor. We designed some initial test circuits, transformed those into our burden monitor strains, and up came some pretty nice results showing that when we induced some circuits, the monitor dimmed and we were using cellular resources. So I think that's pretty much the development of the monitor itself.
Cool. So actually you guys had some really nice results. I mean, literally as the burden monitor increased, increased burden, you see more output, and it's just very, very nice and clean results. But I guess one of the biggest questions I had while reading this was why does it matter? Because when you have increased output, it seems that the burden increases as well. And so, but when our point of engineering these cells is to increase output. So at that point, why do we really care about the burden?
Okay, maybe I can chip in here. Mm-hmm. So, yeah, you're right that of course, if we increase the genetic expression of the synthetic genes that we put in the cell, we will reduce the capacity that the cell has, or we increase the consumption of cellular resources. But the question can be formulated that way, and that's what inspired basically the line of research and the line of thought that we took. Is it true, or is it possible to have different designs for which the amount of output or the production rate of these synthetic genes is the same while using less resources? So you can look at that like an optimization problem. So when you try to manufacture something, what you want to do is to manufacture your product at the lowest possible cost. And that's basically what this is about. So we in the paper, we show that by carefully choosing the dials that we change in the synthetic circuits that we design and implement, we have different impacts in terms of the capacity of the cell, the number of ribosomes, for example, that are used to produce and to express the synthetic genes. And so there is a kind of compromise. And this is very interesting because as we grow the complexity of the synthetic circuits that we try to build, we want to make sure that the initial paper that Tom and myself read about and that inspired this whole research, this kind of global feedback effect between the synthetic circuit and the cell does not completely destroy the behavior of the initial design. And so for this reason, we wanted to make sure that we have simple design rules that allow us to say, look, this is the best way that you can actually implement your circuit in terms of resource consumption, while maintaining a given amount of desired synthetic circuit output. And that allows you, for example, to have one circuit working in the cell, and since you still have capacity, you didn't use your resource unwisely for this circuit, you may still have the possibility of adding another one, and these two still work in the same cell. Whereas if you don't do that carefully and the cell resources are completely depleted with the use of one single circuit, well, you're done, basically. Any perturbation or any addition of an additional circuit will basically prevent the cell from doing its job. Growth will stop and the cell will probably die. So that was basically one of the key reasons. We look at it really as an optimization problem.
Mm-hmm. That makes sense.
And just to add, like it's not just. I mean, for us, we love it because it's engineering and the use of maths optimization in these things. But it actually has some really proper implications for industry. So there's a whole industry out there whose way of making money is they make a protein in very large amounts and sell this for ridiculous costs. So, and I heard a story last week from someone who'd looked at our paper. They said, oh yeah, at Genentech, where they make like over $1 billion out of just producing an antibody from E. coli and getting it in higher yield, someone worked out a trick that increased the kind of the capacity and the growth rate of the cells during this production. They get like somewhere like a 10% increased yield in their production, but because this is such a humongous market that was worth so much, the guy who came up with this got promoted to the board.
Wow. So clearly it's a problem that has scope in industry as well. So kind of related to what you guys were talking about, like clearly this is a very interdisciplinary project. There's analogies made to engineering, but how realistic are these analogies? Like in your little description, you talked about how it could be like thought of as your light bulb dimming if the washing machine turns on downstairs. But I mean, incorporating a circuit even in the genome of whatever chassis you're using still has like implications on how the cell works. So I mean, I don't know, is this analogy really that realistic between the electricity and the biological processes here?
Well, I just want to point out that that analogy doesn't really work in the UK because we have a better strength of electricity, right? So we have like 240 volts here, so when you turn on the washing machine, your light's just fine. The only time I've ever seen this being a problem was when I was in Boston in this old house, and it would really dim. And that can at least teach us things, like, so if you could run your E. coli with a higher capacity with more free ribosomes, you wouldn't see this dimming, right? Because that's the analogy we see here. So that's kind of useful, but I agree that there's some limitation on how far you can take analogies. I don't know, Francesca, if you want to...
I was thinking that in my past research, I was working in synthetic biology, and starting in synthetic biology, I heard a lot of people, a lot of engineers talking about cells as machines, and that you can like guide to do whatever you want, right? And when I started testing the expression of my constructs, I realized that was not really always the case, right? Because we have context dependencies. So NRBS works in a context, and then it changes a lot. And in a way, this project is something that I really like, because it goes in this direction in a way in terms of trying to understand better how cells behave. And so it's not just a simplistic way to look at them, but try to understand which are the needs, if you want, of the cells, to engineer them and obtain what we want in a way that is also compatible with their behavior.
I would just like to add, I mean, of course, these are just analogies, but for any system that has limited resources or capacity, I mean, it's always the same problem. You can see another analogy is with embedded electronic systems. For example, lunar modules that you send up, right? I mean, they have a certain battery life, and if you want them to accomplish their tasks properly, you need to use, this battery power that you have very wisely. So this kind of problem of how you use your resources when you have limited amounts of it is basically coming up in various systems. And I believe that actually, although the cell is a completely different medium, the same fundamental principles are still at play.
You've no idea how many different things we've used as analogies. I mean, it's the same in all the synthetic biology. But discussions about capacity and burden, we've discussed. I've had discussions with people on how do airplanes work out, how many passengers to book, and what do they do if like one airport goes down, and how do UPS do their deliveries? What is the optimum strategy for Amazon in terms of warehouses, right? So, capacity and burden is in any complicated system. So we just look at for the analogies that make the most sense.
That makes sense.
But
so,
hopefully the paper will convince that actually, some of these principles still hold in biology.
Yeah, so actually, like, thinking about that, like, what are some of the major drawbacks that perhaps weren't discussed in the paper, because it is a pretty short paper, that you guys maybe encountered or that researchers should be aware of when using this tool?
Francesca, you want to say?
Yeah, if you mean, the monitor itself, I don't see a drawback of that, apart the fact that you have to implement it in each strain you want to use. But so that I think is the only thing that is a limiting thing.
I think the GFP as well.
Yeah.
Obviously, if you want to have your test circuit, if you want to have an output that's GFP tagged or something, then yeah, it's not going to work with this. But that's we kind of acknowledge that from day one, and I think there are other fluorescent proteins that are available, etc. So that is a limitation, but I don't think it's a major one that you can't get around. I think it's I think as a tool, as an approach to this problem, I think what I think is really good about it is its simplicity, how easy it is to implement. We designed it from day one so that it would minimize all the other problems. We didn't want to go for something that was overly complex, overly sophisticated. We just wanted something that was very simple and could be used by everyone, because we kind of recognized, people were talking about it in the literature, people were talking about it at conferences, wherever we were, it was kind of like the elephant in the room. What is going on with burden? So that was so key to the way that we designed this, was to make sure that it could be used by everyone and to minimize those drawbacks.
Of course, if you want to go to use, for example, other cells, other organisms, you will have to optimize the design of the monitor again and try to insert it again in the genome and play with it. But so there are a few things that.
Yeah, no GFP, no kanamycin, right?
Yeah, kanamycin.
The construct itself uses those.
Mm-hmm.
But other than that, it's very kind of easy for people to make use of this. So we're even now in talks with, have you heard of the company Transcriptic?
No.
So Transcriptic are kind of like this, cloud-based laboratory in a way. Like you code up your experiments and you send them stuff, and then they have robot platforms which do it for you at a small price. So we're just at the process of sending our strains and some of our samples to run on their robots, so that we can see if this assay can be run just for for $20 or $30 at a time for the various different constructs. And we're hoping in the future that maybe this could be like a service that people could send things to them and then run the protocol, pay the money, and then they get the readout of what the burden of their construct is.
That could be very useful, actually. So do you see this this sort of tool working in more complex cells as well, like mammalian cells and other eukaryotic cells? Because I think in an E. coli chassis, things are just a lot easier in general. So I know in the model you mentioned that it's open to any biological system, but well, first of all, how realistic is the model in its approaches with more complicated systems, and also what was your experience and thoughts on that?
So you have several questions there.
Yeah.
I will just jump in quickly with the model, and then probably others can chip in for the other parts of your question. Well, obviously, this question about how good is the model, does it have good predictive ability? These are questions that we faced during the review of the paper itself, and we actually designed new experiments to convince people that indeed the model has good predictive ability, and it gives you results that match up quite well with what we measure in the lab. This was a question that we had from the beginning because the model is quite simple. We only take into account a translation and a free ribosome pool, and try to see what, how ribosome during the translation initiation and translation elongation process are captured by these pieces of mRNA and stay on these before they produce the final protein, which is directly linked to the capacity problem. The more you put on the mRNA before a full protein is produced, the more resources you use inefficiently, if you want. So, yeah, I mean, the model is simple, but I think it is working quite well. Of course, there are many things that we don't take into account, but the goal was to have something that is quite bare minimum, yet allows us to explain the data that we see. And we have other projects ongoing that try to actually exploit this knowledge, even in E. coli. So I'm not going to answer the mammalian question, but even for simple bacteria like E. coli, one of the questions that we're trying to address now is, okay, now that we can measure burden, can you use this information actually to have things that automatically adapt when you have burden? So when you put something in, well, can they directly adapt to the kind of burden that they impose on their host cell while keeping their function?
Let's see.
I think another point to add there really about how relevant the model is and what are its true predictive abilities. So the model and the experimental work were very much done sort of separately. Like conceptually, the experimental work was designed. We built the monitor, we built the test circuits, etc. And the model was sort of built from first principles. So there were very much two separate parts which came together at the end. We ran some simulations through the model, and we got a set of results. We compared those to what we saw in the lab, and they were strikingly similar. And so obviously our first reaction was, wow, this is incredible. Second reaction is, okay, is this a fluke? So what we then did was obviously go back and had to convince ourselves and obviously convince others that it wasn't just a fluke. And so we, as Guy pointed out, we designed some other experiments to really test how robust the model was. And so ran those experiments, and yeah, we kept coming up with the same sort of things. The model was qualitatively predicting what was going on in the cell, which for us was we found incredibly powerful, and you can sort of see those results in the paper. In terms of its applicability to other organisms, I think the model is based on, as Guy said, the translation, and so we're mainly concentrating on the fact that in E. coli, ribosomes are very much a limiting factor. So if you move into other organisms where this may be less the case, then that might not be true so much. It might be post-translation, it might be transcriptional factors that are your limiting factors. So the model might need to be expanded, but I think we're pretty confident that this could be applied through other prokaryotes, and certainly applicable, at least if extended, to other organisms, because the principles that underlie the model and underlie the process and what we're looking at in terms of shared resources, I think that's a pretty ubiquitous approach.
Sure. So does anyone want to add more about the viability of the system, not just the model and other types of cells?
I mean, I was thinking that, it's known from literature that, for example, some mammalian cell lines experience burden when you want to produce some interesting molecule, right? Like, for example, the CHO cells, they're used in industry for the production of therapeutics. And people have seen that they slow down the growth, and there are toxicity effects on them when people try to produce something using them, right? So, this doesn't mean that the monitor is like you can move it straight away from E. coli to mammalian cells, but gives us confidence that there is something similar that is going on in mammalian cells, right? And this could be investigated and needs to be understood well.
So actually, one of the results that I was surprised about, maybe because I don't know too much about translation machinery, is it was actually super surprising that when you just changed the strength of the promoter region, it like didn't affect burden as much as when you changed the ribosomal binding site. And I mean, you kind of touched upon this right now with talking about the translational pool resources, but do you have any other insights onto why this might be the case, or yeah?
So I think the result you're talking about, and this was kind of shocking to us as well in a way. It's quite counterintuitive. When we initially started the project, before we even had the burden monitor, what we were thinking would be that with the available kind of dials you have for changing gene expression strength, then it would make a lot of sense to have like a low copy plasmid, a weakish promoter, and then a very strong RBS, because then this effectively means you can get X amount of protein without having to produce as much DNA and RNA to get there, because everything goes in on the RBS. And we were a little worried that this might cause things like noise. But we thought that cells would grow better that way, because it's just the protein step. And then actually the data came back, and the modeling as well, which actually the information from the modeling almost came first, were giving us the opposite, that when we have like the higher strength promoter, and especially the higher copy number of plasmid, but a weaker ribosome binding site, we get this X amount of product, the same amount, but we get better growth and less burden. So that's completely counterintuitive, because that means for X amount of protein, you need to produce more DNA and more RNA to get there. So why does that cost? Why does that impact the cell less? And so what's great about the way this project came together is the modeling really points out what the problem is, and that is that you can, with strong ribosome binding sites, particularly the ones synthetic biology use, you can do something that is starting to get traction in publications at the moment, which is called over-initiation. So this means like you are initiating too much for your transcript. You're loading up too many ribosomes, and they're loading up at a speed which is greater than the speed in which they can go through the mRNA, produce the protein, and then being released. So you're starting to now get traffic jams. Reasonably controversial in the literature, because direct evidence of traffic jams can only be seen using processes such as ribosomal profiling, which Jonathan Weissman's group have done some really good work on. But all of their work is done seeing if there are ribosome traffic jams on natural sequences within E. coli, and they're not, or they're very rarely seen. But the reality is, if you look at the ribosome binding site strengths of normal genes that E. coli deals with, if you put them into Howard Salas's RBS calculator, they come back with scores of like 2,000, 3,000, 500, 800. Synthetic biology, and particularly like kids with iGEM, are always going, yeah, we want lots of stuff. So they go to his calculator and they go 150,000, right? And this is like, boom, you're turning the dial all the way up. And E. coli never does this with its own genes. It's way, way lower. And so we're having this system. So fundamentally, just to kind of summarize it, what's happening is over-initiation with strong ribosome binding sites, causing traffic jams on the transcript. That means that there are ribosomes on the transcripts, because they're stuck in traffic jams, that are not free to go do the normal things ribosomes do. Mm-hmm. They've been clogged up somewhere in the system. So again, sorry to use another analogy, but in your head, think of like all of the cars in your local city, and if there was a traffic jam somewhere, some of those cars, maybe they're like police cars, they're stuck in the system, whereas they could be off doing their various tasks and solving crimes around the city.
No, that makes sense, I think. And I guess it's still like a area of research that's being studied, since you mentioned that there's some controversiality in publications. So I guess kind of moving it in a different direction, what's next for this project? Like what are you trying to do in the future with this, either this system or expanding it to see other indications or other quantifications in the cell, or yeah, I just want to hear about what's next.
Well, the biggest thing was doing all the extra tasks the reviewers wanted, right? So that that in particular, Francesca and me spent a good half year or like 4 or 5 months of non-stop, everything else off on hold whilst we solve all of these things. But they gave us some good extra tasks to do. Francesca, why don't you say what the kind of stuff you're doing now is?
Yeah, actually, yes. I mean, this monitor is really great, but it gives you like a picture of what's going on in the cell. It doesn't give you the details, right? I mean, it doesn't tell you who is playing the part inside the cell. So who are the molecules that are triggering the burden response inside the cell? So what I'm doing now is going from this bigger picture to the details inside the cells and looking at the transcriptional changes that the cells experience when they are burdened by the devices, right? And then we want to try to understand which are, using different devices, which are the common players of this burden response in the cells, and try to understand how to use these to better tune the expression from our synthetic devices, so something like that.
And so, as part of what Francesca's been doing, we've already got some RNA-seq data, which looks at what happens 15 minutes after you start something that causes a burden, half an hour and an hour later. So we see all sorts of changes going on in the expression of the mRNAs of the cell in response to ours. And we see some interesting things. Our own plasmids, the ones from the paper that cause the burden, they can, within an hour of inducing expression from them, they can be taking up 30 or 40% of the transcription of mRNA in the cell, right? So they're almost getting on par as the same as all of the rest of the mRNAs, which is kind of even more shocking than that. One of the things we spot is that if you change the level of the RNA in the cell from your system, the cell doesn't change its growth rate too much. It doesn't care about that. All it cares about is whether you're using up the ribosomes or not.
Mm-hmm. And on the modeling aspect, is there also some future work that you'll be working on, or is it kind of a done project?
No, I mean, we are currently exploring actually whole cell models, so taking into account more aspects beyond just translation, involving the use of other resources, essential resources of the cells, amino acids, energy, transcriptional resources, translational resources, and trying to build up from there a kind of platform that we can use to actually in silico interrogate basically the behavioral interaction between circuits and host cells. And so one of the key aspects for us going forward is not only to know how the host cell responds to the insertion of synthetic pieces of DNA, but also to design things or synthetic circuits that use this information in a kind of feedback manner to actually regulate their own behavior, their own synthetic gene expression, depending on the amount of burden that they impose on the host. So automatically reacting to the global state of the cell. To actually have a system that is more robust and automatically adjusting the demands, the pressure, the burden that it imposes on the cell, so as to keep things in check.
We'd be really interested as well in seeing what other people do with this. Now it's out there and it's reasonably simple. Mm-hmm. So, for example, from iGEM, Randy Ratberg, when I discussed this with him about a year and a half ago, said, for all of the characterization people want to do, and they don't want to have to then work out a way to then get a GFP output, can they characterize whether their constructs are on or off and doing things and how much expression they're making by looking at how much burden they cause? So the decrease in GFP expression rate of the cells over the hour after they've induced their system could then infer how much expression their system is causing, even though they don't have an actual readout of their system. It's kind of like a way you can characterize things without having to attach them to a reporter protein. We're not doing that. Someone do that for us.
No, I think definitely it's a tool that would be very useful for any synthetic biologist out there, because I mean, I've had brief experiences with synbio research, and you always see your cell growth decrease, but you don't know why. And I really think this, the direction Francesca is going as well, with looking at more detailed reasons why, and then also with the modeling, trying to get the cell itself do some feedback control of your gene expression and stuff. I think that's a really, really useful direction.
Yeah, we had a paragraph in the paper that ended up getting chopped because of the word limit. That kind of puts a bit of caution on it that there will be growth rate defects that are specific to your system. So if you're expressing a protein which uses up energy, just it has some reaction which turns over ATP or something like this, then this is going to cause a growth rate defect specifically because of the task of this protein. Or like a membrane protein which misfolds, and you're going to see some burdens to do with that. So what this is really trying to capture the burden that is general for everything, which for everyone's different designs is only going to be a subset of the real burden. In some scenarios, it might be nearly 100% of the burden is because of this, just the use of the resources for gene expression. But for other designs, it may be that only a certain fraction of the burden that you see is this specific type that we've been working on.
Okay. And where would the other burden come from? Sorry, I might have missed this one.
I don't know.
Toxicity effects.
Things that you produce a transcription factor which then binds on lots of places around a genome. Something that is. We call those specific burden effects that are associated with what you've produced. And normally that will be the protein part of what you've produced. There may be some scenarios where you produce some RNA part which then binds somewhere and has some bad effect.
Mm-hmm. Okay, well, I think we're almost nearing the end of this scheduled hangout, but if you guys have any last words to talk about this research, or I don't know, your feelings about the future of synthetic biology, or whatever you want to talk about, now's the time.
This paper, I just want to point one thing. Me and Guy debated about this for a while, but we were very happy we did this. We put the paper up on bioRxiv as soon as we got the feeling that it was almost there with the reviewers. So this, I think I spent Christmas Eve getting it ready and putting it up on bioRxiv.
You did.
And that's great. We didn't get any issue with Nature Methods saying, whoa, you can't do that, right? Because it's our preprint. But it meant that we have a version that is open access that everyone can see, formatted slightly differently, so a lot of the detail is kept within. So there's slightly more detail in that version, and it also, gets out there earlier. So although this just came out in Nature Methods this month, it has been up there online for people to see since December, and I really hope that everyone in synthetic biology starts using bioRxiv because it's a really good resource.
The only thing that I would like to say is that it is a real pleasure to be able to merge disciplines in the way that we have been able to do through this paper and in synbiology generally. So I think people from really different backgrounds, from mathematics, from engineering, from biology, from molecular biology, it's a really great asset to learn new things and to discover new things.
Yeah, and Reese and Francesca, do you want to add anything?
I want to thank you for this. It's really nice to have the chance to talk about the things that you have done secretly in the lab for 2 years. And thanks really for this opportunity.
Yeah, I think I'd like to echo that, and I think also what Key was saying about bringing different disciplines together. I think it's been really fun over the last however many years it's been to see how the modelling and what have you can inform the experimental design, etc. And for it actually, for it to be a real part of it and not just a kind of little bit of modelling you do at the end. For those things to really come together is really cool to see. And I think it's hopefully this is an example of how powerful it can be when you kind of really think about how you can use those different disciplines together. But yeah, and to finish up, I think I'll say thank you so much for giving us the opportunity to talk about it. I think I've had a lot of fun with this project. I think we all feel pretty passionate about what it can do, and I think this is potentially the start. We can see what other people go and do with it, because it's it's out there for people to use. It's going on. Where's it going to? You're sending the strains out
to AdGene.
To AdGene. So I guess people will be able to get the strains from AdGene. And yeah, so everybody go out and use it and publish and let us know what you've done with the stuff. It's
It's just the beginning.
Yeah, well, just to give some context on how long we've been working on this as well, right? This project began when me and Reese still thought that the TV show Lost would have a good ending.
Very. That's good.
Yeah.
Well, thank you all for coming here, or being part of this hangout and really creating a very beautiful tool and exemplifying the interdisciplinarity that can come through in research and biology. Like, it's really nice to see this, and you have some really great results. So even if it took a really long time, I mean, clearly it was worth it. And yeah, thank you so much.
Thank you very much.
Thanks.
Cheers.
Thanks.
Bye-bye.